doesn't work very well, but it is viable. Now, the size of the genome that they have is 7.7 million base pairs of DNA. So just about 10 million base pairs of DNA. To put that in context, baker's yeast or brewer's yeast, I still haven't gotten my beer yet. That's about 12 million base pairs of DNA. So this is a genetic engineering operation on the scale of baker's yeast. So you can begin to think about engineering something as big and as significant and as important as baker's and brewer's yeast. Here's a project from France. This one really impressed me. The human genome was sequenced in 2000, our own DNA, and people have now, you can get that sequence on your computer and you can study it, right? So if you want to reverse engineer stuff, there's a nice thing to reverse engineer you. These folks in France found that there was basically genetic fossils, pieces of DNA that appeared to have been inserted in our own genome about 5 million years ago, whatever our ancestors were 5 million years ago. And these pieces of DNA stopped working. They came from what looks to be a virus, and then they all sort of evolved independently, one from the next. They fell apart in different ways. And so today we have about 20 or 30, I can't remember the exact number, copies of this ancient virus lying dormant in our own genome. What they did is they analyzed the 20 different broken copies, and because they were each broken in different spots, they could infer what the ancestral virus was 5 million years ago. So they reconstructed that, and they put that back into tissue culture, meaning a controlled laboratory environment, and that produced a fully infectious human retrovirus. So it's like the movie Jurassic Park, but without needing to have the physical sample of the amber, if that makes sense, because the information's already inside you. I'll come back